Is this fine? Everyone can hear me. Hello. Thank you, Trevor, for the introduction. So as Trevor said, I'm going to be talking about the design of an HIV-1 envelope trimer for the isolation of novel broadly neutralizing antibodies. So, as you may know, HIV-1 is a global health concern. And in last year alone, 1.3 million new infections were reported. Um, and so the search for an HIV vaccine remains prominent. However, due to the high mutational rate of the HIV virus, this is still quite a challenge. The main antigenic target of developing HIV vaccines is the HIV-1 envelope protein situated on the surface of the viral particle. It is a heterotrimer that is highly variable between HIV-1 strains and subtypes and is the most immunogenic part of the HIV virus, presenting multiple antibody epitopes. This is the first crystal and cryo-EM structures of the gold standard highly stable HIV-1 trimer called BG505. Initially, it was thought that X-ray crystallography provided the best resolution of these proteins. However, nowadays, cryo-EM is more widely used as it is not reliant on the production of crystals and is thus not limited by the flexibility and complexity of these proteins. These structures have also assisted in identifying multiple antibody epitopes, as shown in the structure over here. And these trimers have been designed based on HIV-1 envelope proteins from viruses that are mainly moderately neutralization resistant. And in that, we have isolated multiple antibodies and broadly neutralizing antibodies that are currently being implemented in clinical trials. So, as mentioned earlier on, broadly neutralizing antibodies are actually rare antibodies that develop in a small subset of HIV-1 infected individuals. And these antibodies are able to prevent infection through binding to conserved epitopes found on the HIV-1 protein. However, recent studies have indicated that there is an emerging resistance to existing BNAbs by circulating HIV-1 strains. And so, a potential solution to this is the isolation of novel broadly neutralizing antibodies. However, using HIV-1 soluble trimers that are based on the envelope proteins of highly neutralization resistant trimers. So, these envelope proteins will be inherently resistant to existing BNAbs and thus allow for the isolation of novel BNAbs, which may in fact neutralize Anyone else have any jokes? Yeah, I'm gonna try now. Okay, cool. All right. So, um, difficult to neutralize isolates used within the study were selected from a previously funded IRV study, which actually screened 183 HIV viruses against a panel of 6 broadly neutralizing antibodies. And these antibodies actually targeted 3 of the known BNAB epitopes. So from the 183, I selected the 5 most difficult to neutralize isolates and screened them against a panel of 18 different broadly neutralizing antibodies. And these antibodies targeted the following corresponding epitopes of the HIV trimer. And as you can see, where you have gray, it means no neutralization has occurred. And where you are seeing the warmer colors, it means that neutralization has occurred. And so I selected the 3 most difficult to neutralize isolates and found that they actually were resistant at quite a few of the epitopes. I then used those 3 isolates to screen participant plasma. All the participant plasma in this project came from the Caprisa cohorts, which collected participant samples from HIV-1 infected women over several years in the early 2000s in Durban, South Africa. And out of the 20 participant samples screened, we found that only 2 were in fact able to neutralize the fresh 1388 isolate. And of the 2, the CAP287 participant seemed to be an ideal an ideal candidate for the isolation of BNAbs using the fresh 1388 soluble trimers. And so I then designed 2 iterations of the fresh 1388 soluble trimer. The first iteration just contained your usual stabilizing mutations, and the second iteration contained an additional 2 proline substitutions, which was said to increase the trimer yield as well as stability. And so in moving forward, I then started characterizing the 2P soluble trimers and found that when doing an ELISA, there was in fact high binding of V3 loop antibodies. And this is not ideal as this indicates that the trimer is either in the open conformation and not exposing BNAb epitopes, or that the V3 loop is in fact overexposed. And this is not ideal for B cell sorting, as this would heal B cells that are specific to and have antibodies that are non-desirable and they're not broad. And so we wanted to move this trimer from being, having an overexposed V3 loop or being open into one that actually has a more compact V3 loop. So this led to the design of 3 different V3 stabilized mutants, which had mutations in the V3 region that were said to increase hydrophobic packing. I then reassessed the binding of the 2 antibodies that bound to the V3 loop and found that all of that the binding of both antibodies to the V3 mutants was reduced in comparison to that of the wild type. And so I selected to move forward with the 316W trimer. However, I needed to confirm that this trimer was in fact in the closed conformation, as when these trimers are in the close conformation, it presents BNAB epitopes. And so, as you can see, the 316W trimer is concordant with that of BG505, which is our gold standard, and it's found mainly in the closed conformation. It was also able to create a 3D reconstruction of this 316W trimer. And all of this work was done in collaboration with the UCT EMU, thanks to Dr. Jeremy Woodward and Claire van der Berg. And so, in confirming that majority of the trimers were stable, I then knew, okay, I could use this trimer as a B cell sorting bait. I was also able to collaborate with the EBIC unit at Diamond, and thanks to Dr. Jeremy, was able to create a 3D reconstruction of my 316W trimer that was comparative with that of a previously published structure of BG505. I was then able to sort 252 B cells from the Caprisa participant CAP287 using the 316W trimer, as well as a cocktail of stable HIV-1 trimers. And we were also interested in a group that was, in fact, specific to both the 316 trimer and the previously stabilized HIV-1 trimers. From this, I then began the antibody isolation process using the rat-pig methodology and screened my isolated antibodies using a neutralization assay. I tested these antibodies against 5 different viruses. And obviously, where you're seeing the red, it means that these antibodies were capable of neutralization, and then the gray, obviously, no neutralization occurred. So this results indicate that CAP287 may in fact have broadly neutralizing antibodies, and hopefully we can isolate some of them soon. And so, in conclusion, new BNABs are required for HIV-1 vaccine strategies and therapeutics due to the emerging resistance to existing BNABs. The characterization of the 316W soluble trimer revealed that it was, in fact, stable and native-like, and I was able to sort 252 B cells. If I should isolate any novel BNABs, I hope to determine the epitope specificity using cryo-EM. I would like to say a huge thank you to everybody at the ERU unit for their support, especially my supervisors. Thank you to all of our amazing collaborators at the UCT EMU, Caprisa, and EBIC. And thank you especially to all of our funders and the Caprisa participants. Thank you so much, everyone.
Thank you very much, Danielle. That was a pretty good talk. Okay, so does anybody have anything to either question Danielle about or discuss with her? Where do you think she should go next in her work? Not for travel, in the sense of the development of the project. Yeah, Jeremy.
Yes. Do I?
Yeah.
It's bringing back. So I think this is probably an obvious one, and it's probably what Danielle's going to do next. But but I think it would be very interesting to see the antibodies bound to the envelope protein, so you can see, exactly that interaction. I mean, do you have plans to do that? I mean, you'll have the antibodies, and you'll have the.
Yes. So right now we're in the process of actually sequencing them, and I hope to get those results within the next coming weeks to hopefully see if we actually have something that is in fact novel. And then if it is so, we will look at doing antibody conjugation to the trimers, and then hopefully with your help, actually sending it over to EVIC for another round of cryo-EM. Yeah. Thank you so much.
Thank you so much. Good luck.
Thank you.
All the best. Okay, so the next